Hi everyone, and welcome to this video lecture about the biology of marginality and psychoneuroimmunology. Quite a mouthful. In today's video lecture, we're going to cover some information about the impact of chronic stress and some neuroscience information that helps to ground that discussion a bit for you to understand why it is that perpetual stress ongoing tends to have such an impact on us. I, of course, am missing our time together today. I'm noticing it and thinking about you all and hoping you're having a decent break on this Monday. So let's review some key terms that we're going to cover today. We're going to learn about gene transcription, gene translation, ribonucleic acid, what's known as messenger RNA, and ribosomes. These are all functions, parts of epigenetics and the way in which your genetic kind of coding is basically understood and translated into proteins. Then we also will learn about some issues around gene expression, for example, methylation, and talk about your epigenome. We'll talk about a little bit about apoptosis. I probably will be fairly brief about that today. We're going to focus most of our time around chronic stress and what happens with chronic stress. And there are certain key terms you should be familiar with. Some of these are extra than the chapter, meaning the chapter kind of alluded to them, but there's been more information come out even since the chapter was written that you should be familiar with. And so, for example, you may not have read about concepts such as hypercortisolism, the attenuation hypothesis, and the metaplasticity hypothesis, but we will still be focusing in on those today. We'll also learn about inflammation and what happens in the inflammatory process and talk about cytokines and the big eaters, macrophages. So the flow for the next 2 weeks, this counts as 1 week. Next week, when we meet again, will be the second part. We'll be learning about the impact of stress. There are two major ways in which stress impacts us. There is chronic ongoing stress, and there is traumatic stress. These are often confused, and it's important, I think, to clarify what we're talking about here. Both processes involve a release of cortisol and adrenaline to a person's system that is perceived as being problematic, that causes issues. Traumatic stress is in response to a threat to one's direct survival or direct health. And so you think about this as, for example, being the victim of a violent crime, being in a war-torn area, something like that was really extreme. Chronic stress does not have to be as extreme as that, but it tends to persist for much longer. Whereas traumatic stress, usually you're thinking about one or two isolated incidents that still have a really strong impact on a person, getting into a car accident, for example, or an experience of sexual assault, something that has a prolonged influence on a person. Whereas chronic stress would be things like ongoing racism, ongoing homophobia, it could be bullying at school, those issues that can persist for longer and can have an impact on a person just because of the perpetual nature of the stress. So we'll learn about today the impact of chronic stress, and we'll learn about traumatic stress next week. Sometimes you'll hear these referred to as big T and small t trauma, with traumatic stress being big T and chronic stress as being small t. So we're going to answer some big picture questions today. What is the role of heredity and environment in development? To what extent do genetics inform the development of personality traits, character, and mental health conditions? What is the role of stress in mental health conditions? And as you will see, it's a fairly large role. And then what is the biology of marginality? Meaning, how does it impact us to experience things like marginalization, or oppression, or discrimination, or prejudice? To explore these questions, we need to review the basics of cellular biology and epigenetics. Now, I have worked hard to try to cull some information from this slide deck for today so that we just cover the most important information, so I'm not rambling on and on in a video lecture. And hopefully, a lot of this information won't be too overwhelming. If you're interested, you could read a book by Alberts called Molecular Biology of the Cell. Most of you will have no interest in reading this, but if you get particularly jazzed about the deep down cellular level, this would be a book to check out. Okay, so let's learn about cellular function and epigenetics. So we've learned about neurons. We've learned about the neurotransmission process, for example, by which a cell communicates with another cell. The electrical charge passes down the neuron, all that stuff we've learned about before. Today, what we're going to learn about is how do cells kind of continue to live, thrive in the state that they're in. How do they develop new parts of the cell, for example, new proteins? What parts of the cell are transcribed, from your genetic material is transcribed to help support the cell? You should know that the human body is largely composed of cells and fluid. Sounds really comforting, doesn't it? We're just bags of cells and fluid. Well, we're obviously a lot more than that. It can't be reduced to that. But in terms of what we're made of, that's pretty much the gist of it. Cells perform different functions. There are different types of cells, which they complete through receiving nutrients and converting them into energy. One example of those cells is the neuron, the nerve cell. Here's an example on the right-hand side of a hippocampal neuron, meaning this would be a neuron found in the hippocampus. It's interesting, right? You imagine neurons to look different than that, but you can see there that they have a lot of dendrites that really do spread out quite a bit and spread out on both sides of the cell, if you will. Look almost like, I don't know, branching like from two different sides. And then you can see the soma in the middle. That's the cell body. And then you can see there the axon. The axon connects the dendrites with the axon terminal buttons. And of course, you don't see the axon terminal buttons in this diagram, but at least you get a sense for how spread out the dendrites are in a cell. Here is a very kind of straightforward diagram to depict the basic structure of the human cell. You have the nucleus in the middle, the membrane around the nucleus, and then you have things within the membrane, such as mitochondria, such as the cytoplasm. The main reason that I show you this is because within the soma of the neuron, for example, the soma being the cell body, you have a nucleus. That is true for all cells, and it is within the nucleus that we have DNA. Okay, now you're wondering, what on earth is DNA? Ready for this? Deoxyribonucleic acid. Try saying that one 10 times fast. DNA contains instructions for developing new proteins. And DNA is very important to us because it helps the cell to thrive, to be able to, for example, develop proteins to recover from injury, or develop proteins to, to address a new need it has in its environment. The instructions that DNA contains are known as genes. Note that only 1% of DNA contains genetic information, or is protein coding. Now, what we mean by that is 99% of DNA does not include these instructions for developing proteins. It's kind of inert, really. But it's the 1% of DNA which contains genetic information that we're interested in. Okay. The DNA sequences that code proteins are known as exons, whereas the sequences of DNA that do not code proteins are known as introns. Okay. Introns regulate information about protein production. What we mean by this is when that protein coding occurs, how it occurs, and how much protein is produced. Now, if you're anything like me, you're reading this and thinking, why do proteins matter? Like, what's the deal about proteins? Why are they so essential? Well, we'll learn about that in just a second. The entirety of your DNA, all of your DNA, is called the genome. Okay, so basically all of your genetic information, if you were to have that all collected together, that would be called the genome. And note that humans share 99% of the same genes, and we're obviously not all the same. And so there is obviously that 1% that makes us unique in who we are. But it's interesting that we do share a lot more in common than we do separate. RNA, ribonucleic acid. This is all background information, by the way, to understanding the impact of chronic stress. If you're wondering, why are we learning this? It's important for later on. While most genes contain information for creating proteins, there is a small percentage of genes that produce what's known as ribonucleic acid, or RNA, instead. Okay, so remember, only 1% of DNA is genetic. It contains genetic material or information called genes. And of those genes, not all of it contains basically protein coding genes. Some of it contains stuff that creates RNA. Okay. Now, what's the deal with RNA? Well, RNA is a similar related molecule to DNA and works to code, decode, and regulate the expression of DNA. You can think about it as being like the meta version of genetic material, where it basically decides how much protein is going to be produced, what kinds of genetic material is going to be used to produce those proteins. So like DNA, RNA is a nucleic acid that's also found in the nucleus of a cell, the soma. RNA can also be found in a cell cytoplasm. You see it in mitochondria. That's above and beyond what you need to know for this class. During gene transcription, RNA polymerase binds to DNA, which then unwinds so it can be copied. This is a very short sentence that unpacks a lot of information, so let me break it down. You probably remember that basically there is a lot of information included in DNA, and it has to be wound up tight so that basically it's otherwise it would be way too long of a shape. And so it is bound up by what is it bound up by to make chromosomes? It's bound up by histones that coil around it to make chromosomes. And chromosomes basically are tightly packaged DNA, and you need to unwind them in order to basically transcribe information from it onto RNA that is then translated. This is a complicated process that I have a diagram for in a sec. But that's basically, RNA is helping to take the genetic material and then make protein from it. Okay. So it transcribes or copies information from DNA to then build amino acids into chains that eventually become proteins. Here's the diagram I promised you. You see the middle, the kind of center circle there. That contains DNA. You see the double helix kinds of strands. And basically what's happening is that RNA helps to unwind the tight package of the chromosome and then basically will copy information from genetic material onto itself, and then it will move outside of the nucleus into the cell body and into outside of the nucleus. And then it is that information on the RNA is then transcribed by ribosomes that basically create amino acids. You see them forming into a chain there, and eventually that becomes a protein. So there's a lot of steps involved, but the key idea here is that there are processes by which your genetic information gets actively kind of translated, transcribed into, and made into proteins. So that's the whole reason you have DNA, is to make proteins. Okay. Now, what the heck, right? Proteins, that's it? I'm just a bag of cells, fluid, and proteins? Well, let me explain. There are many different kinds of proteins. You're familiar with many of these, and they have very important functions. One type of protein is antibodies. They protect the body through binding to viruses and bacteria, stop you from getting sick, reduce infection, things like that. Enzymes read genetic information and are responsible for chemical reactions within cells. They are therefore very important to the whole chemical reaction process, such as what happens during neurotransmission. Messenger proteins, such as hormones, like, for example, growth hormones, think about brain-derived neurotrophic factor, or BDNF as an example of this, coordinate processes between cells and tissues, such as creating new synapses. Very important. So in other words, proteins have an essential role in fighting off infection, in making sure that neurotransmission is occurring, and also in creating new synapses, all very important to cellular functioning. Now, all well and good, right? But you're wondering, okay, what does that relate to chronic stress? Well, what's really interesting is that the process I just described, by which your DNA is transcribed into proteins to to enhance the functioning of the cell. That process is very influenced by your environment. And to understand that, you have to turn to the field of epigenetics, because there are other chemical compounds that can attach to segments of DNA. And when they attach, they can influence which parts of your genetic information or material is transcribed and translated and which is silenced. For example, methyl groups can bind to a segment of DNA and are known to stop the production of proteins from that segment of DNA. In other words, they stop, they curtail, they silence gene expression. In other words, they stop the coding and development of proteins based on that genetic information. When chemical compound alterations affect protein production, we call this epigenetics. And it's important to note that things like methyl groups attach to your DNA because of environmental influences. These are things that do not occur because of just basic genetics, birth genetics, that are unrelated to your environment. They happen as a result of your environment to help you ready for this, respond to your environment. Okay. The entirety of the genome and the chemical compounds which influence gene expression comprise the epigenome, which is basically all of your genetic information and all of the tags, all of the chemical compounds that attach to your genetic information to determine whether or not those genes are expressed or silenced. So finally, we get to some more applied information about, okay, well, what does this mean to me? So let's talk for a minute about epigenetics and methylation. Remember that methylation turns off gene production, stops gene production, okay? Suppresses gene expression. There's a YouTube video that's worth watching if you're really interested in this, and I'll include that in the slide deck that I send out. So what's happening with methylation is it basically stops certain parts of genetic information from being transcribed and translated. And you'll see when we get to cortisol, that methylation process is very, very important. Okay, so a lot of information I covered there. Here are the key points to be familiar with. You have all of us have genetic information. Not all of it is translated, transcribed into proteins. There are processes that influence whether or not that gene information is basically is used to develop proteins, turned on or turned off, not used to develop proteins. And that process is very influenced by your environment and methyl groups, things like that. The chemical compounds that attach to DNA strands have a large role in determining whether or not your genes are expressed or not. Okay, now let's learn about stress. One of the ways that I want to engage you is by asking you to reflect a little bit. Now, obviously, you're going to have to pause the video to do that, but if you feel comfortable, pause the video in just a couple secs and ask yourself, what do you notice in your own body when you experience high levels of stress? What have been the impacts to your physical and mental health of that stress? Pause for a second and just think about that. So I'll respond for me personally. When I'm stressed, I feel it, right? I feel the tension in my muscles. I feel myself getting sometimes shaky or having sweaty palms, or any of that process of sympathetic activation that you've been learning about. And it can have quite a significant impact to my physical and mental health. There have been times in my life when I've been very stressed. I was bullied from about age 6 to 16 or something like that. And during that time period, I was a wreck, right? I was very depressed. I basically would be shaking a lot of the day, particularly in certain situations where I think I was going to get bullied. It just had a profound impact on my own health. And I think that's true for a lot of us who experience chronic stress, that it just has this prolonged impact on us and it makes life miserable. It also can have physical health impacts that we'll be learning about later on. Stress is not all bad. We have to also acknowledge that. There is an adaptive nature of stress. You've probably heard some of these terms. If you haven't, eustress versus distress. Eustress is helpful, useful stress, whereas distress is not helpful when it becomes too much. The Yerkes-Dodson law talks about how you develop eustress versus distress in regards to optimal performance, and this is how it goes. Let's say that you have an exam coming up, easy analogy. If you are totally low on the stress scale, as a 0 on the stress scale out of 10, you may experience some negative effects from that. You may not study very hard. You may not review information like you're meant to. You may not take the test very seriously. You may turn your attention elsewhere. If you are too high on the stress scale, though, if you're a 10 out of 10, you may feel shut down, unable to concentrate, can't remember the reviews that you're doing, can't retain information. So obviously neither is ideal, right? And we want something in the middle. And that's where eustress comes in. It's enough stress to get things done, to have some oomph, to have some energy, but not too much stress that we feel basically incapacitated. And that's, by the way, a good model for healthy sympathetic activation, right? No stress at all. And a person who feels numb, if you will, to their environment, that's not healthy. We need to be able to adapt to stress in our environment, but in a way that doesn't shut us down. So I've talked a bit about how low stress and high stress affects our behavior there. So is cortisol always unhealthy? The answer is no. In small contained doses, stress can be beneficial to attention, to motivation, to processing, and to overall health, including immune responses, believe it or not. So in other words, a small amount of stress can actually help you respond to your environment in a productive way. And for example, if you have an infection in your system, there should be an inflammatory response to that to help control that infection. If your immune system is compromised and you don't have that response, you're in trouble. So it helps when we have a moderate amount of stress, our bodies and minds to adapt to the environment, and also warns us that changes need to be made to our internal or external environment. And that's the messaging that we then respond to. So for example, if we have an exam coming up and we start feeling stressed, the message is, I need to study, right? I need to get into gear. And if you don't receive that message, that could be problematic. Or if you receive the message, though, that's too strong and it sends you into overdrive, that's also not helpful. Let's learn a bit about cortisol, and I promise that'll help us eventually lead back to methylation and understanding what's happening during chronic stress. Cortisol is a hormone associated with the mobilization of energy resources, especially in response to stresses in the environment. We've talked on a very kind of grand scale about stress in our environment and sympathetic activation and what's required to respond to our environment, right, to stress. What we're going to learn about now is, again, the small details of what's happening and cortisol's role in preparing us for action. It does some really important things that mobilize us, prepare us for action. It increases blood sugar, giving us more energy, especially to the muscles. It enhances metabolism of carbohydrates, fats, and proteins, again, burning through, if you will, energy storage to get us ready. And also suppresses the immune system. And you're probably wondering, well, how's that helpful? Well, during a period of stress, you don't want your body to be digesting a ton of food. You also don't want it to be fighting off a ton of infection. You want it to be all of the blood, if you will, to be mobilized towards large organs and muscles. And so that's the reason why you tend to see suppression both to the immune system and also the gastrointestinal system. This helps a person conserve and use energy for important tasks facing them, such as responding to threats in your environment. The typical trajectory of cortisol production during the lifespan is kind of interesting. There is a steady increase from middle to childhood to early adulthood, and then eventually levels off from adulthood onward. So in other words, you don't feel the same sense of urgency, motivation, sometimes stress overall as you get older. In the psychology literature, we often talk about as people age, they become less neurotic, but they also become more conservative, interestingly enough, if you look at the ocean model, if you know what that is. And there is some degree of neuroscience support for this, that basically the highest amounts of cortisol or motivation or recruitment of energy reserves to face our environment is experienced during middle childhood to early adulthood, and eventually there's a leveling off. Cortisol levels are highest in the morning for us throughout the lifespan, fluctuate throughout the day, and taper off at night. So in other words, cortisol also has a role in getting us ready for daily tasks, right? It has to do with alertness, for example. And you may remember that I said that before that we are diurnal, not nocturnal animals. And so that pattern makes sense when you also understand our overall build, if you will, as creatures. So this is a graphic that helps you understand cortisol secretion release throughout the day. You have 2 sleep cycles there, just to kind of give you a sense for what's happening. Alertness there, you can see, rises throughout the day. You become most alert, believe it or not, around midday. And for those of you who are not morning people, that'll make very good sense to you, and then eventually drops off towards bedtime. Your body temperature also changes. Your body temperature is highest when you're awake. It actually drops at nighttime during sleep. In terms of growth hormone, believe it or not, it is highest during sleep. And that's important because, again, if you're not achieving adequate sleep, that has roles in, for example, the birth of new neurons and synapses. And so that's why we often attribute lack of sleep to things like depression. You see cortisol at the bottom there. Cortisol, again, is highest in the morning and slowly kind of, with a bit of an up and down pattern, declines throughout the day until bedtime. So in other words, you are, even though your alertness is not at its peak, you are at your most motivated, if you will, when you first wake up in the morning, and it starts to sag as the day goes on. We've talked a bit about the HPA and SAM axis already, so I'm not going to belabor this, but just as a brief reminder or refresher, remember that cortisol is released from the brain, for initial messages sent from the brain by the hypothalamus to the pituitary gland and eventually to the adrenal cortex just above the kidneys. So let's talk about what happens with cortisol again quite quickly. Once the thread is passed, a message is sent to the hypothalamus from the adrenal glands to discontinue production of cortisol. At some point, you want this cortisol tap shut off, because without that, you're going to continue to be ready for action. And over time, that's going to cause problems. For example, you don't want your immune system suppressed forever and ever. That's not good. You don't want your gastro system suppressed forever and ever. Also not good. So you also, for example, don't want dilation of blood vessels forever and ever. All those things will cause problems if they last too long. So in other words, some degree of activation during a stressful event is okay. It's just our natural method of addressing the stressor. But if it continues too long, it causes these negative effects. Okay. Now, if we are able to re-regulate, return to baseline, not be activated based on parasympathetic activation, whereby basically parasympathetic activation is about how the adrenal glands send a message to the hypothalamus to turn off sending CRH or corticotropin releasing hormone down to the pituitary gland, which then releases ACTH, adrenocorticotropic releasing hormone. You probably remember all this stuff from earlier, that then releases cortisol. There's a process for that, right? And there's that tap is shut off by the hypothalamus sending a message to the adrenal glands. And again, this re-regulation or return to homeostasis is called allostasis. Allostasis is about this sense of kind of balance, that you get activated, but then you also are able to recover from activation. Here's a graphic that describes what typically happens and what the problems happen when there's too much ongoing stress. On the left-hand side, you see the balance, right? The up and down of stress, the seesaw, where you get activated and eventually there's a return to balance, okay? On the right-hand side, you see allostatic load. That's a term that means too much stress, getting out of balance, where you basically have too much stress on the sympathetic branch of the ANS that's releasing too much cortisol, and you're stopping the return to parasympathetic activation by stopping the body from turning off the tap, if you will, from turning off the cortisol tap. So let me try to explain this a little bit in the next example. when we talk a little bit about the Meaney Szyf experiment, and then help you understand what's happening with methylation and why this return to parasympathetic recovery is not happening with chronic stress. So you're probably maybe a bit confused. Let me give you a 30-second recap to make sure you're hanging in there with me, okay? So what I've talked about so far is that stress is normal, stress is okay. The problem is if it persists too long. And there are reasons for why stress persists too long. One of the major reasons is that, and by the way, when I'm talking about stress persisting too long, I'm talking about cortisol continuing to be released in the system. I should be clear about that. One of the reasons why cortisol continues to be released is that the body eventually stops the tap from being turned off. It stops the re-regulation. It stops parasympathetic recovery. And to understand that, you need to be exposed to the Meaney Szyf experiment, and then to just further kind of explore what's going on, that cortisol re-regulation, the stopping of cortisol release, is being halted. And that's therefore the cortisol tap remains on and continues to be emitting cortisol for long periods of time. So in the Meaney Szyf experiment, it's a very famous experiment, you've probably read about it before. There were two sets of rat mothers based on behavioral observations. Group A had mothers who were attentive to their young, demonstrated nurturance through licking and grooming behaviors, which in rats is, kind of a nurturing behavior. Group B consisted of mothers who did not provide the same licking and grooming behaviors, meaning did not demonstrate nurturance to their young. Okay. The researchers, because of course this is an animal study, you know how I feel about animal studies, don't love them, but here we go. They impregnated both sets of mothers and they switched the offspring at birth. A bit Machiavellian perhaps, but for this experiment, important. And this is what they were interested in. Basically, were the rat pups who were born to mothers who were not nurturing, but raised by mothers who were, did they experience stress greater or less than rats that were born to nurturing mothers, but were switched and were raised by the non-nurturing mothers? Because if we can understand that, then we can figure out to what role is genetics versus environment impacting things like stress response. Okay. To do this, the researchers took measures of whether genes that control stress hormone production were creating proteins or silenced via methylation. So I want you to pause for a moment and ask yourself, which set of rat pups do you think had a dysregulated stress response system? Do you believe it was rat pups with possible genetic predispositions towards greater stress response who were born to mothers who were non-nurturing, who were then raised in a nurturing rather than stressful environment? Do you think they had the greatest dysregulation? Or do you think it was rat pups who did not seem to have genetic predispositions, meaning they were born to mothers who were nurturing, but were raised in a stressful environment? Which of those two do you think was most dysregulating as a condition? Pause for a second and think about it. Well, the answer is interesting. What they found was that rat pups born to attentive mothers but raised by inattentive mothers had an increased stress sensitivity compared to rat pups that were born to inattentive mothers but raised by attentive mothers. Those rat pups found no increase in stress sensitivity. All of this leads us to conclude and hypothesize for humans, and there are some studies that support it, as you'll see, that it is environment that has the greater influence on whether we develop chronic stress responses, much more so than genetics. And our environment, therefore, is very important to attend to. In other words, we are not doomed to be stress sensitive. It is that our environment can make us so. And environment means the way we were raised, but also the larger environment around us, the social environment. And I say this because, I have a very wonderful colleague who I do some consulting with, who does a lot of work around racial trauma and healing from racial trauma. And they talk about, look, when they go to trauma trainings and they talk about ongoing stressors in a person's environment that could be traumatic, such as racism, that a lot of presenters will dismiss that out of hand as, that doesn't quite fit their model. for how to work through trauma and stress, because most models have to do with realizing that what happened is in the past, that the traumatic event is in the past. Well, what happens if the stress persists? It's not in the past, it's ongoing, and we haven't developed great models for how to work with that. We're just developing those now, really. And I'll note that there is nothing in the DSM, believe it or not, that diagnoses an ongoing stressor-based disorder. We don't have a PTSD variant for chronic stress, and there are many of us who want that in the DSM. So pause for a moment. Do the results surprise you? Why or why not? Did you think it would be genetics or environment that would be the greatest influencer on stress sensitivity? Next, we'll examine what implications this has for humans. So here's what we know in human studies. Early life stress in human children causes the same issues. It causes methylation, which we reviewed earlier, in the same glucocorticoid receptor gene related to the production of stress hormones. What you have to understand here is, remember, methyl groups and how they can silence gene expression and protein production. Well, what's happening here is that prolonged stress turns off a person's engagement in parasympathetic recovery, so that the stress continues on and there isn't a recovery from it. A phrase that I love to use with clients that a colleague once told me about is that anything non-nurturing can be traumatic, and ongoing can cause issues that really impact a person, because a person becomes, as you'll see, sensitized to stress, and that becomes a major issue for a lot of kind of cascade effect, as you'll see, for other issues. Oh, we're getting right into it. So now that you've learned how chronic stress works, how, for example, the ongoing release of cortisol works and the methylation process involved in that, you should understand what chronic stress means, and we'll kind of address quickly where it comes from. It can result from numerous factors: acculturation, bullying, discrimination, marginalization, living in poverty, toxic relationships with a partner, with family, toxic work environment with, for example, a boss or a co-worker who's really difficult and awful to be around. I have a client right now who got out of a job with a boss in which it felt traumatic to be around that person. And also violence and exposure to violence. Those things aren't necessarily directly traumatic in the classical sense of post-traumatic stress, meaning they don't affect whether or not a person is going to die, right, or going to have grievous physical bodily harm. They may, of course, but often they have more of an emotional effect, and it's that emotional effect that causes this, this related to this chronic stress response. You're probably familiar with ACEs, or adverse childhood experiences. There are 7 types of adverse childhood experience in the original ACEs study that came out in '98, and they talked about the following as examples of chronic stressors, if you will: psychological abuse or emotional abuse, physical abuse, sexual abuse, substance use by a household member, depression or suicide attempt by a household member, violence towards mother or stepmother, and imprisonment of a household member. In the study, the ACEs study, they basically found if you had 4 or more of these factors, you were at much greater risk of having a slew of symptoms, of effects of this. So here they are. Basically, they found if you had 4 or more of these stresses, it doubled or even tripled the risk for the following in the individual who grew up in that environment: alcohol and drug addiction in adulthood, cancer, cardiovascular disease, depression, obesity, sexual promiscuity defined as 50 or more sexual partners, sexually transmitted infections, and suicide attempts. Now, obviously, there are a lot of other factors that would cause a person to experience these things, but it is important to note that early childhood stress, if perpetual, if ongoing, has major risk factors, and we have to address that as a culture, as a society. We have to address that clinically with kids who are going through chronic stress situations. We have to be able to help adults understand what happened with them and help them reorient and engage in parasympathetic recovery as best they can. And you'll see more about my recommendations for that towards the end of today's slides. This is a complicated pathway, but a very important one to understand. Kind of really summarizes some information we've talked about and adds some additional pieces. So what happens what is the process by which a chronic stressor, bullying, prejudice, racism, homophobia, those issues, how they cause eventual mental health symptoms and physical health symptoms? That's at the end of the pathway. Well, what happens is you have methylation of genes, or the suppression of genes, that's associated with HPA axis regulation. That has to do with cortisol release, adrenaline release, and that leads to chronically high levels of cortisol because the tap is shut off. Basically, it's not allowed to be shut off, so cortisol continues to be released. This leads to other effects, and we haven't talked as much about these yet. Cell overactivation and degradation. Cortisol does cause impacts at the neurological level. It overactivates cells, particularly through inflammation and cytokines, which we'll talk more about. It reduces brain-derived neurotropic factor and glutamate, which again is associated with the birth of new neurons and synapses. It leads to chronic inflammation responses, which cause other major issues. Cytokines, for example, overactivate neurons and can lead to the death of neurons. And then you have immunosuppression. So as we mentioned before, that cortisol naturally suppresses the immune system, and over time that can cause illness. So all of these things paired together, if you look at the next step in the pathway, lead to long-term depression, meaning the death of neurons over a period of time, and therefore volumetric reduction in CNS brain structures. That also, this is the awful thing about this pathway, will then result in even less CNS control over the HPA axis, and even more what's called hypercortisolism, even more chronic cortisol release. So it's like this perpetuating cycle of cortisol release. And you may be wondering, well, why is the body doing this? It's doing this because the environment demands that we address it, that we respond to it. So a kid that grows up in a, for example, high crime, high poverty environment in which they're a person of a minority background, and they experience, for example, police harassment, as well as outright racism from those in their community. Over time, that person has a lot of stresses they have to respond to, and the body and brain must respond to that if the person is going to survive, right? They have to be ready for action almost at any moment. They feel on edge constantly because of this. And you can't fault the body for that, right? The body's trying to help the person address those stresses. What you can fault is the way that we have built up our society so that some people experience that, because over time that has a major impact on physical and mental health. Here is the metaplasticity hypothesis that's related to hypercortisolism, which I've gone over just a moment ago. You have a persisting stressor that leads to methylation or silencing of genes, then chronically high circulating cortisol, reduction of brain-derived neurotropic factor. Apoptosis is cell death, programmed cell death, that I don't want to go in too much into, so I think it'll take us away from the topic. Then you have long-term depression in structures that regulate the HPA axis, and then eventually the person reaches a new homeostatic set point by which they're used to high circulating cortisol, and that further weakens control over the HPA axis. That's called the metaplasticity hypothesis. In other words, we respond, we adapt to our environment in a plastic kind of way. That's what we mean by neuroplasticity. We're adapted to our environment, and that causes a person to have chronic cortisol responses. This hypercortisolism has a link to depression. This has been called the most consistent and robust biological finding in psychiatry to date, the linkage between HPA axis dysregulation and major depression. An estimated 50% of major depressive episodes are linked to chronic stress and hyperactivity or overactivity in the HPA system. In children, chronic stress is a risk factor for subsequent first episodes of depression, and methylation of genes associated with suicide attempts in adolescence. So in other words, depression, suicide attempts, these are all related to high stress environments. Depression itself, by the way, is also associated with an 8 to 10% reduction of hippocampal volume and therefore may be a risk factor for later neurocognitive disorders such as Alzheimer's. And so we worry about the long-term impact, even beyond depression, which in itself is enough of an impact. Now you may be wondering, well, okay, can you share with me a study that kind of shows this from beginning to end with actual people? And there is a very useful longitudinal study by Trickett and colleagues from the University of Virginia that did this, and I want to talk through that study. It's one of the very few longitudinal studies into the relationship between stress and epigenetics in humans. So in this study, Trickett and colleagues studied children who were and were not exposed to early childhood sexual trauma, sexual abuse, over the course of 23 years. Okay, and they had two groups, one group that was exposed, one group that was not exposed. They had a very high retention rate, 90% retention, which is very difficult in longitudinal studies. They also had 50% of their sample was, I believe, people of color. So they had a very strong representation of diversity in their sample. To understand the impact of abuse on the HPA axis, participants were given corticotropin-releasing hormone, or CRH, which is, of course, associated with eventual cortisol release. Okay, because we talked about CRH, ACTH, eventually cortisol gets released that way. So they were given that, and then basically to see to what extent was cortisol going to be released when that process was initiated. Now, here's what they found. During childhood, females with abuse histories had increased ACTH production, increased basically cortisol release in response to the CRH compared to the control group, which you would expect, right? In other words, when kids who have and have not experienced sexual trauma are compared with their stress response in a typically stressful situation, okay? Children who've experienced chronic stress, those who've experienced sexual assault, sexual abuse, those children have, even to normal stressors, have an exaggerated stress response. Okay, but here's what's super interesting. The process was different in early adulthood. The opposite effect occurred. Females with abuse histories had reduced ACTH production compared to the control group. The opposite occurred. This is called the attenuation hypothesis. And basically what happens here is that a person becomes so acclimated to high levels of cortisol that they need a lot of cortisol, a lot of CRH, if you will, initially released to generate any kind of cortisol. In other words, they're so used to high stress in their environment, it takes a ton of stress to feel stressed at all. And so that's also a problem, right? That you're not responding to stress in your environment because you become numb to it because you're so used to high stress. It's almost like the bar has been raised so high that you really require a ton of stress to get over the bar and to get into a stress response cycle. Also called the asymmetrical stress response. I think I've described this pretty well already. So other study findings were notable here and also supported what you've previously learned from the ACEs studies. Females with abuse histories had higher rates of teen pregnancy, were twice as likely to re-experience sexual assault or victimization as adults, twice as likely to experience domestic violence in adulthood, had a 4 times incidence of suicidal ideation and self-injurious behavior, had higher rates of preterm delivery, had higher rates of obesity, and we'll talk about why that is. Do I talk about that today? If I don't, basically the adipose fat tissue is influenced by cortisol, that when you're in a stress state, there's more development of adipose fat tissue. And it also accelerated pubertal development and began, the females began hormonal and sexual maturation sooner as a response, because cortisol also plays a role in the HPG axis, or hypothalamic pituitary gonadal axis, relating to hormones that pertain to development, to sexual development particularly. So I say all this because you can see here that there are real-world implications to a person, even beyond their physiology, and their physiology probably plays a role in some of this, for example, adipose tissue. One of the things we worry a lot about is the transgenerational influence or inheritance of this trauma of the chronic stress. And what we know is that epigenetic modifications, for example, attachment of methyl groups, remain even after cells divide and can be inherited similarly to DNA, which was referred to as transgenerational epigenetic inheritance. Thus, a person's environment can affect not only their current functioning, but also the functioning of their offspring. So in other words, you can inherit a chronic stress response. Now, that can also be turned off based on how your environment responds to you. But it's important to note here that a person, if they're born into a high stress environment, their parents have already primed them to some extent to be ready for that environment. Okay, we've covered a lot of things so far. Let's just kind of recap. We have covered the process by which genes are expressed or silenced and how that impacts things like chronic stress response, such as the tap being turned off to cortisol for it to continue to flow and to develop this hypercortisolism, and how that has major impacts to physical and mental health, as well as other behavioral impacts. Now we're going to learn about what happens to the person's immune system. This is called psychoneuroimmunology. It's related to stress. Immune cells, white blood cells known as leukocytes, have receptors for cortisol, believe it or not, and chronic cortisol prohibits their functioning in response to infection. That's what we mean by saying that the immune system is suppressed. Inflammation becomes more intense during chronic stress. So let's talk about PNI, or psychoneuroimmunology, in chronic stress. There are two principal mechanisms: activated immunosuppression, which we've talked about before, increases susceptibility to illness. It can also, by the way, lead to cancerous cell development and promulgation, which is why you tend to see high rates of cancer in people who've experienced, for example, adverse childhood experiences. Inflammation also becomes more intense, resulting in cell death because of cytokines. Remember, cytokines overactivate cells and destroy them. This causes things like nerve damage, increases susceptibility to arthritis, can cause autoimmune diseases, Crohn's disease, ulcerative colitis, and also fibromyalgia. So ongoing inflammation is no good. With Muna, you may remember the case that we've been exploring, stress could potentially worsen her ulcerative colitis, and you would want to be addressing her stress response as part of your work with her. Inflammation by itself is not problematic, just like stress and cortisol is not problematic. It's crucial to immune system function. What happens during infection is that you have white blood cells or leukocytes that are attracted to the area, that kind of recruit blood flow to the area. And that leads to fever, and fever in itself is not bad. You're raising body temperature to try to control and basically kill the infection. And what happens is that cytokines are related to that. They recruit blood flow to an area, and then macrophages, or the big eater, I like to talk about the pack person here, basically come in and swallow up the external agent, the virus, for example, that's not meant to be there, to destroy it. Okay, so that's how we fight off infection. That's how we engage in preventing infection. What happens, though, over time, without needing to kind of get into this graphic's probably a bit too much for right now to get into, is that if you have too much inflammation, it's going to cause problems. Let me just skip the slide, I guess. It causes damage to cells and tissue. And the reason for that is that cytokines cross the blood-brain barrier and activate the HPA axis. They make you stressed. This can alter neurotransmitter metabolism and reduce BDNF and glutamate, which, remember, is associated with the birth of new neurons and synapses. This can result in depression, manic states and bipolar disorder, non-response to antidepressants, and physical health conditions such as cardiovascular disease and diabetes. No good. Now, what do you do about all of this? Well, there are things that you can do that stop you from having that cascade of problems that start from chronic stress. And here's the things to consider. Physical exercise and sleep are very important to your overall response to stress. And believe it or not, counseling itself is not sufficient. In fact, there are no studies that have found that counseling in and by itself leads to the reduction of cortisol and leads to parasympathetic recovery. Instead, there are other other kind of ways to help a person recover, and physical exercise and sleep are the way forward, because we're especially interested in helping a person redevelop those neurons and synapses that have been lost because of hypercortisolism, chronic stress exposure. Physical exercise is very important, as is sleep. By the way, in the UK, at one point or another, they were allowing physicians to give prescriptions for people with depression to go to the gym for a month. I wish they were still doing that, and I wish it had translated to the US. In the US, of course, we just have medication and counseling as options. A single bout of physical exercise increases brain-derived neurotropic factor, a single bout. Ongoing physical exercise increases gray matter volume, meaning birth of new neurons from a gray matter is unmyelinated, in the frontal lobe and hippocampus, and improved white matter integrity in children. That's the myelinated neurons. Those neurons have, they're just better able to function. They have the nutrients that they need. Okay, the BDNF, the glutamate helps with that. Sleep is also related to BDNF production. And so if a person is sleeping right and exercising right, that can have major impacts. One of the other things, of course, to work on is to help a person engage the parasympathetic system almost as a muscle by doing a lot of deep breathing throughout the day. We talk about this in a couple of weeks, but if you can get a person breathing at the rate of, say, somewhere around 7 to 8 breaths a minute, and kind of slow, regular, rhythmic breathing throughout the day, that helps a lot with parasympathetic recovery. The other major thing, of course, to address here, before we kind of leave you with 2 reflection questions, is that you also have to advocate for changes to the person's environment. You have to make sure that the chronic stresses are being reduced, because without that, stress will continue because the stressor is still there. So, for example, if a person is experiencing bullying, you have to address that, help the client to address that. If the person is experiencing ongoing racism, you have to be part of efforts to start reducing that in communities and be an anti-racist. Those things matter to people and help a lot with preventing these problems from continuing and from developing in the first place with future clients. So we'll learn much more about advocacy in your social cultural class. For now, this is just kind of a point, kind of something to take note of, that advocacy matters, not just working in the room with the client. So we've covered a lot today, a lot about the stress response system and the way in which that influences people. One of the questions I want to leave you with, a couple of them, is how might this information inform your practice as a counselor? What are you thinking about that? How does it change how you think as a counselor, how you might be working as a counselor? And then what aspects of your counseling approach might you change based on this information? We are at time, everybody. I hope that was helpful in covering some of the most important information about chronic stress. By the time we meet next week, we'll pick it up and talk about traumatic stress writ large, post-traumatic stress, and see what happens during the process of acute trauma. If you have any questions, feel free to email me. Otherwise, I will see you all next Monday. Take care, everyone. I'll see you then.